Welcome to section 2.20, where we're going to discover the chloroplast. So as our intro to photosynthesis, we really need to talk about the organelle that's going to do this, at least in all eukaryotic cells, plants, algae. With prokaryotes, they'll have these thylakoids, but they really don't have the rest of the structure because they don't have membrane-bound organelles. So with the chloroplast, let's go over some of the basic parts. So we have a chloroplast envelope, which if you remember from the nuclear envelope, means a double membrane. So we have this outer membrane and we've got an inner membrane, neither of which do that much for us right now, so we're going to largely just say they exist and move along. The ones that we're going to care more about is the stroma, which is essentially the cytosol of the chloroplast, so it's like it's cream filling. So this will be the liquidy section, and this is where the second half of photosynthesis will take place, namely the Calvin cycle. We also have the thylakoids, which always remind me of kind of like hollow pancakes. If you had pancakes with like liquid inside, I guess maybe if you could fill them somehow with like strawberry filling, except in this case it'd be cytosol. And so they've got stacks of these, lots of these, and each of these kind of pancakey looking guys is a thylakoid. The key thing about them is they have that large membrane, even though they're kind of pancake shaped, they have a lot of membrane. And the thylakoid membrane is where the first part of photosynthesis takes place, which will be called the light reactions. So it will play an important role. Don't just kind of write it off because it looks weird and looks like a pancake. So structures, just once again very quickly, outer membrane, inner membrane, both of which we're not going to discuss too much. The stroma, and you can see here it's just the liquidy insides. That's going to be where the Calvin cycle takes place, the second part. I'll just write it in a different version. And then we're going to have stacks of these thylakoids are called grana, but you'll see that we'll have these thylakoids that will be stacked up, and we have lots of them. And the main goal by having lots is that we're using the membranes that they possess to embed our light reactions, to embed part of photosynthesis. So the more of them we have, the more membrane we have to embed stuff in, the more photosynthesis can be done because we can do more light reactions, and light reactions are needed to do the Calvin cycle, or the dark reactions as they're sometimes called. So we have lots of these thylakoids to help make sure that we're capable of doing enough of the, and I'll just write that in here, the light reactions. Now, we're going to talk a little bit more about just the light reactions. And so this first step here is going to involve something called the photosystems, or you might see it called the electron transport chain, ETC. But these are going to be a series of proteins, both pigments and some other proteins that are more enzymatic, that'll serve various functions, transport, other stuff. But they'll be embedded together in kind of a chain in the thylakoid membrane. So one thylakoid membrane can have lots of these. You know, the more space you have, the more of these chains we can fit. And so if you see someone mention photosystems or the electron transport chain, they're talking about the light reactions. So the pigments will be the guys that absorb these photons, which just means light. So light is ultimately going to provide energy to the pigments. The pigments then will have their electrons get excited. So they'll have these electrons now that are excited or have increased energy. And then depending on which way we're going to go, the photosystems are able to either take these excited electrons and take their energy and stick it onto this electron carrier called NADPH. So this is a fairly simple process. Make sure these electrons that are high energy get stuck on this molecule to now make it a high energy molecule. Or we can do a slightly more involved route where we go through and take the energy from the electrons and use that energy to make an H plus gradient. So we're going to use it to pump H pluses into the thylakoid. So if this is our thylakoid, we're going to pump a bunch of H pluses in, so they're smashed in there together. Now they don't want to be in there together smashed in. They would prefer, and if diffusion occurs, they would want to leave. So we're going to allow them to leave, but we're going to make them leave through a special enzyme called ATP synthase, which acts kind of like a turbine. So as these guys leave, it's like me allowing you guys, kids, to leave the room, but I make you go through one of those cool revolving doors. And so as you leave, you give up a little bit of energy to spin the door. And I can use that energy from spinning the door to do something. In this case, ATP synthase, this enzyme, will use that energy to build ATP. And so you can see here, this is the inside of the thylakoid. This is the outside, the stroma. And so there's a lot of H pluses in, and by letting these H pluses go out, it allows us to go through and make ATP. And so this H plus gradient can be used as essentially an energy source to allow us to make ATP. Much like we can use a waterfall, letting the water do kind of what it wants to, go to a lower energy, it gives off energy in the process. So if you have a dam or a waterfall, you can use it to spin turbines and create electricity. In this case, we're making it spin a turbine that's like a protein turbine and use that to make ATP, so we can store energy. And so our end products of the light reactions are ATP and NADPH. That's really what we're after. We'll discuss some other stuff where we split water. There's some other things that occur, but we'll focus more on that in the next section because section 2.21 is really going to focus just on this, just on the light reactions. So we'll go a little bit more in depth. And then step 2 is going to be the Kelvin cycle, sometimes called the dark reactions because it doesn't directly need light. It needs the end products of the light reactions. It needs ATP, it needs NADPH, or namely you can say it needs energy, because it's going to take CO2 and the remnants of splitting water essentially, so specifically H pluses are what we're after. It's going to take these guys and stitch them together to make a sugar. Now initially it's going to make a sugar that's going to be C3H6O3, otherwise known as G3P or glyceraldehyde 3-phosphate. Because that name is kind of terrible if you're trying to remember something, what you'll see is a lot of people just assume that we're going to stick two of these together, and so that would give us C6H12O6, which we would call glucose. So if you see them talk about that photosynthesis or the Kelvin cycle makes glucose, it doesn't really. It makes essentially half a glucose, a G3P, each time you do it. But because you can stick two together and get glucose, and glucose is more user-friendly, a lot of people will use that instead. But keep in mind, it can also take this G3P, this initial tiny sugar, and it can also convert that into lipids and proteins and nucleic acids and every other molecule that's an organic molecule that the cell needs. So don't assume that photosynthesis is purely about sugar. It's largely about sugar. It's one of the more common things that you'll make as a plant, but it can also make the rest of the stuff they need. They can't eat. If they need something, they're going to need to be able to make it themselves, and they can. You know, once they're done, they can finish processing this to whatever the heck they want. It's not a dead end. It's just the beginning of the next part of the plant's life. I hope you guys have enjoyed this. This wraps up our photosynthesis overview, and I'll see you guys soon.